Hello and welcome to another video in our course on cell biology and genetics. In this video, we're going to talk about DNA replication. So what are you going to learn in this video? First, I'm going to explain what DNA replication is and how the whole process works. I also explain why we say that DNA replication is semi-discontinuous and semi-conservative. And we're also going to look at enzymes that are involved in DNA replication. After watching this video, you will also understand the terms replicon, replication origin, replication fork, template, leading, lagging strands, RNA primer, Okazaki fragments, and proofreading. And you will also understand the function of enzymes involved in DNA replication, such as helicase, DNA polymerase, primase, DNA ligase, but also the function of single-stranded binding proteins. By the way, you can find written notes on what we're going to discuss today at medicinecanbeeasy.com. The link is also in the description. Okay, so that's it for introduction, and now let's get down to business. DNA replication is a process in which the DNA molecule duplicates, producing two molecules of DNA from the original one DNA molecule. This is very important when cells replicate, for example, as each needs its own copy of DNA. Now, in eukaryotic cells, DNA replication doesn't happen at only one spot of the DNA, but at several at once. The small parts are called replicons, and their beginning, at which the replication starts, a replication origin. In prokaryotes, the situation is completely different, but we'll talk about that in a different video. Replication starts by the two DNA strands unwinding and separating. This is possible because of a special enzyme called helicase, which breaks the hydrogen bonds between the two strands. Once the two DNA strands are separated, single-stranded binding proteins, SSBs, bind to the two strands to prevent them from joining back together. The structure that forms when the two DNA strands separate is called a replication fork. After this separation, the two strands are ready to be duplicated. They are each going to work as a template by which the complementary strand is going to be synthesized. And for that reason, we call them template strands. The special enzyme that synthesizes the new strands by adding new nucleotides to it is called DNA polymerase. DNA polymerase follows the basic base pairing rules. Adenine pairs with thymine and cytosine pairs with guanine to ensure that the two strands are complementary. The problem is, however, that DNA polymerase cannot synthesize the new strand out of nowhere. It needs a short RNA sequence called RNA primer to know where to start. And this short sequence is synthesized by an enzyme called primase. Another problem is that DNA polymerase can synthesize the new DNA strand in the 5' to 3' direction only. Because the two DNA strands are antiparallel, each strand is going to be synthesized in a different manner. One is going to be synthesized continuously, as the DNA polymerase will simply move in the 5' to 3' direction, and we refer to this strand as the leading strand. The other is going to be synthesized discontinuously in small pieces, because the DNA polymerase has to shift every time that helicase separates another part of DNA to work in the 5' to 3' direction, as you can see here. This DNA strand is referred to as the lagging strand, and the small DNA pieces are called Okazaki fragments. These Okazaki fragments are later connected by another enzyme called DNA ligase. We say that DNA replication is semi-discontinuous, as one DNA strand is synthesized continuously and the other discontinuously. What is also fascinating is that DNA polymerase is capable of proofreading. This means that the enzyme checks that a correct nucleotide with the correct base has been added. This eliminates the possibility of making a mistake, which, considering how long the DNA is quite amazing. One last important characteristic of DNA replication is that because the newly synthesized DNA is going to contain one original DNA strand, the template, and one new strand, we say that DNA replication is semi-conservative. Half of the molecule is old and half new. Okay, so now we'll summarize what we've learned today. First, we said that DNA replication happens at several spots at once, and that these spots are called replicons, and they each have a replication origin. Then we looked at the process in detail and said that it starts by helicase that breaks the hydrogen bonds between the two DNA strands and separates them. Then SSBs bind to the strands to prevent them from joining back together. Primase then synthesizes a primer which allows DNA polymerase to work properly. DNA polymerase can then add nucleotides, but only in the 5' to 3' direction, which means that one strand, the leading strand, is going to be synthesized continuously, and the other strand, the lagging strand, discontinuously. For that reason, we said that DNA replication is semi-discontinuous. And Okazaki fragments that formed on the lagging strand are then connected by DNA ligase. We also said that DNA polymerase is capable of proofreading, which means that there is going to be as few mistakes as possible. And finally, we said that DNA replication is semi-conservative, as the new DNA molecules contain one old strand and one new strand. Okay? Great. So this is it for DNA replication. Thank you so much for watching. If you found this video helpful and you'd like to watch more videos about cell biology or medicine in general, please subscribe. Thank you again, and don't forget that medicine can be easy.